So what we do, because we aren't very good studies for genetics, we do use pedigrees. Plus, actually now you have this we have this ability to do genetic counseling to actually look for certain alleles. There are many alleles that have been isolated, but it's really very much of an arduous task to find a specific allele for a particular trait. But the allele for breast cancer has been isolated, so it does become a good use with regard to this pedigree. And so what the pedigree is going to do for you is, if in fact you have two individuals or a individual that you have no idea what their genotype might be, you might be able to see what they look like in terms of their phenotype, but you have no idea what their genotype might be, that you would go back into past generations to see what's been expressed then, and be able to trace back into what the genotype most likely might be for that particular individual. There is this I guess there is this concept of disease, which we haven't really talked about, and some disorders can be inherited, and most of the disorders that are life-threatening, and they can be life-threatening, what happens is if in fact somebody inherits that disorder and it's life-threatening, it's very unlikely they're able to reproduce. So a number of recessive, or the number of diseases that are inherited are many, don't get me wrong, but the mutations don't lead to somebody who reproduces, because unfortunately they die before they can get to that age. But a relatively harmless recessive inherited disorder, they show up only in the homozygous recessive allele, so only when you have the two little r's, but carriers can be heterozygote. So heterozygous can be a carrier of a trait, and so an example of a disease that is or it's not even a disease or a disorder, an inherited characteristic is albinism. I guess it's actually very dangerous if in fact it's, for example, this alligator, which is in a controlled environment. It's in a national park, or a it's not a national park, but it's in a park in South Carolina. But if in fact this particular animal was in the wild, it wouldn't be allowed to reproduce because it wouldn't live. So sometimes it's not necessarily a disorder that is harmful for that particular individual, but certainly is harmful for its continuing. But albinism is a recessive condition, complete lack of pigmentation in the hair and the skin. And that can happen in many different species. We see it in our own species, but in many species you can have that. And then another recessive allele that remember with the recessive allele you have to have the homozygous, so it has to be double. What you have here is this is a family from Austria called the Habsburgs, and one of the reasons why I brought this in is your book just brings up the fact that generally over time it has been thought of as taboo to marry close relatives. The areas where that did happen are in these royal families of older times. So royal families, say in the 15th century, where they married their very close relative because they wanted the bloodlines to stay very pure, according to their beliefs, I guess you might say. But this did result in a lot of recessive traits being expressed that were not positive for those individuals. And what this picture is showing you is this chin that was exaggerated and created because of the marrying of close relatives. And then I also put this, it looks like a book from medieval times, and I don't know what it's expressing because I can't read it, but put it in there to express that one shouldn't marry a close relative, and that it even came down through many eras. Cystic fibrosis is one of the more deadly diseases, not quite as deadly as it used to be. Very common, and about 1 out of 2,500 people of a certain European descent, so of European descent. And in the United States, there's a huge population of European descent. And what happens is this absence of it's a defective allele, where you have the inability to get rid of chloride ions, and that then it creates a buildup of mucus. It used to be that people with cystic fibrosis only lived to be their early 20s because of respiratory problems. Now their lives have been extended to be almost normal lifespan, and certainly normal lifespan if they can get a lung transplant, if that can happen. Very expensive and not so likely. What we're looking at here is that pedigree and how that pedigree can be used. This comes from your book, and it's also kind of fun because it's very harmless. Because you have this widow's peak, so you have two sisters at the bottom, one with a widow's peak and one without a widow's peak. And the idea, and this is the third generation, the idea is we are trying to find out if in fact the one with the widow's peak has is homozygous dominant or heterozygous dominant. And so you can see if you go back that most likely, particularly if you're going back to the parents of herself, she has a 50-50 chance of being, no, I guess it's a 50-50 chance of being heterozygous dominant, and a 25% chance of being homozygous dominant. And then you have the individual with no widow's peak, and we know that the parents, because of the understanding what the genetic makeup of the two sisters are, we know the parents had to have been heterozygote dominant. And then you can do the same thing with the free earlobes. Widow's peak is dominant. The free earlobe is dominant also, as the attached earlobe is recessive. And you can look around your family members or look around wherever you are, and you can see the variety of whether somebody has a widow's peak, and this is both male and female, and whether somebody's earlobes are attached or free. Sickle cell disease, we talked about this a little bit already, but it's very, very common. 1 out of 400 individuals of African American descent. So pretty common to have at least the trait. It's caused by a single amino acid substitution, so just one. And the homozygous individual, the hemoglobin, so the ability to carry oxygen, is abnormal. And then you can also have a heterozygous individual for the sickle cell trait, and that person will be considered a carrier, and unfortunately that even carrying the recessive a recessive gene with a dominant gene can lead for that person to have some effects, so some of the symptoms. But if a person has a homozygous recessive traits for sickle cell, it unfortunately is a fairly severe disorder. Heterozygotes, as I said, for sickle cell trait are healthy but suffer some symptoms, and they are considered carriers. Um, 1 out of 10 African American people of African American heritage has sickle cell trait. So it's 1 out of 400 actually has the disease, 1 out of 10 the trait. That's a high frequency of an allele. But one of the positive trade-offs, the heterozygotes are less susceptible to malaria. So there's an advantage. And what you have here is you have a nice pedigree, you have a father with a trait, a mother normal, most likely a 50-50 shot that the children are going to carry the trait. And then if you move on down where you have a father with sickle cell, a normal mother, all of the children are going to carry the trait. So you can see how that 1 out of 10 with regard to sickle cell trait is going to amplify quite quickly. Some disorders are caused by dominant alleles. So it's not always the recessive allele that's causing the disorder. A disorder that is caused by a dominant allele is achondroplasia. It's a form of dwarfism. What happens is the epiphyseal, the capacity for the bones to grow in length, particularly the appendicular bones, the bones of the arms and legs, have been is shortened. The time that they can grow in length is shortened. So the individual usually has a fairly normal torso, but longer or shorter arms and legs. But this is caused by a rare dominant allele. And then with regard to, as I said, with regard to most of these inherited disorders, most of the individuals don't live long enough to reproduce, so that actually causes an end, I guess you might say, to that disease. But there is one, Huntington's disease. The disease doesn't actually manifest till the person is past the age where they would actually probably have offspring. So no effects until 35 to 40 years of age. And it actually is a very, very sad disease, causes a deterioration of the nervous system. So somebody who has a pedigree or has a family history of Huntington's disease very, very likely will want to go through genetic testing, perhaps, to see if they have, to see what their potential, what the potentials are with regard to passing that on to their children. And the way, one way that genetic testing is actually prior to actually becoming pregnant between the male and the female, but the genetic testing might lead the mother and the father to go to other avenues other than genetically trying to create a child. There may be other avenues for them. But if in fact a person, and we do tend to do an amniocentesis, or people tend to opt for an amniocentesis. What this is cells are taken out of the fluid that the baby is floating around in, so out of the placenta. And so skin cells are constantly being sloughed off, and so those cells the fluid's taken out and the cells are looked at, and they're looked at for the entire genetic data of the baby. Certain diseases can be identified. Not all things can be identified, but certain diseases can be identified. And then the advantage of the amniocentesis is done, say, week 14, 16, something like that. The advantage of the chorionic villus sampling, the CVS, a sample of the placenta is removed, and the advantage of that, and then looking at the karyotype, is it can be done in week 8, 10. So it can be done a little bit earlier in the pregnancy. Both are useful techniques to try to see if in fact there's any kind of genetic issues with regard to the child. And we will pick this up on the next chapter.